This video lecture is on Rhesus blood group system. The Rh blood group system is one of the most polymorphic and immunogenic system known in humans. It is the most complex system with over 45 antigens. That is why it's called polymorphic because it has 45 antigens. But of these 45, there would only be 5 immunogenic antigens, which will be discussed in the incoming slides. Discovered in 1940 after work on rhesus monkeys, thus the term RH, in recognition of the rhesus monkey as the laboratory animals for the discovery of your RH blood group. RH gene, located on a short arm of chromosome 1, is the gene that is coded for the formation of the RH antigens. The RH antigens are highly immunogenic. The D antigen is the most potent. In the previous slide, it was stated that there are 45 antigens. Of these 45, these are the 5 highly immunogenic antigens of your RH. The uppercase D, lowercase c, uppercase E, uppercase C, and lowercase e. So this is in arrangement as to highly immunogenic to rarely. And this is the reason why it's only D that we detect in the red cell suspension of an individual if we are to phenotype the RH grouping of an individual. For the reason that among these 5, the D is the most potent and it can specifically react with the use of its anti-sera, anti-D. Exposure to less than 1 ml of Rh positive red cells can stimulate antibody production in an Rh negative person. So this statement would mean that the antibodies for Rh are immune antibodies, meaning that they are only formed upon stimulation. Unlike your ABO, antibodies are naturally occurring. But for Rh, it is an immune antibody which is a product of stimulation coming from the D antigen exposure. The exposure could possibly be due to incompatibility in transfusion, meaning transfusing an Rh positive blood to an Rh negative recipient, or incompatibility during pregnancy, where mother is Rh negative and the developing fetus is Rh positive. On Rh genetics, there are 2 closely linked genes controlling the expression of all Rh antigens, and we call them codominant alleles because they would really be expressed in every haplotype. Like the RHD gene that determines the expression of the D antigen, while the RHCE gene determines the expression of the uppercase and lowercase c, same with uppercase and lowercase e antigens. For Rh nomenclature, there are several different systems of naming or nomenclature that theorize the inheritance of the Rh system. And these are your Fisher-Race, Weiner, and Rosenfeld, the 3 most common among the several different systems of nomenclature for your Rh. The Fisher-Race nomenclature, or we so called it CDE terminology, this is the most commonly used among the 3 by the WHO. This is developed by Ronald Fisher and Robert Race of England. Thus, the theory on Fisher-Race named after the developer. They theorize that the Rh antigens are controlled by a complex of 3 sets of genes with closely linked loci, and these are your DCE, they are codominant gene, complex codes for DCE antigens. So this is an illustration of the gene coding for Fisher-Race. The D gene, uppercase D gene, produces the D antigen, while the lowercase d gene means you have the absence of the D antigen. For uppercase C gene, then that codes for uppercase C antigen, and same with the lowercase c gene, codes for the lowercase c antigen. Uppercase E gene codes for the uppercase E antigen, and lowercase e gene codes for the lowercase e antigen. You notice that there is no antibodies made mention in the nomenclature for Rh because they do not possess naturally occurring antibodies. Antibodies, again, I would like to emphasize that they are immune antibodies product only by stimulation during exposure to the D antigens. So there are 8 gene complexes at the Rh locus for Fisher race. Fisher race uses actually the DCE order. In other books, they would use the CDE. CDE just to follow the alphabetical order or alphabet order that C comes before D and D comes before E. But originally in the Fisher race nomenclature, it made use of the DCE. Starting with the D, since D is the most immunogenic of the five common or immune this is the most potent rather of the 5 immunogenic Rh antigens. So you have here 2 columns. On the first column, all are in uppercase D. So which means that these are all Rh positive individuals. When we categorize Rh phenotype of an individual, we always look at the D, if it's uppercase or lowercase. If it is uppercase, then it denotes that the individual is Rh positive. On the other side, you have a lowercase d, which denotes that these individuals would have an Rh negative as its phenotype. So these are the combination, possible combination of the gene for Fisher race for the Rh positive, and this would also be the combination of gene for the Fisher race for the Rh negative. Okay, next would be the Wiener theory. In Wiener theory, there are 5 major Rh factors or antigens. We have the longhand version. If it is RHO, then that denotes for the uppercase D, which means that the individual is Rh positive. If it is RH, then it denotes uppercase. All you have to check is for the presence of the single prime or double prime. If it is single prime, then that denotes for C. If it's double prime, then that denotes for E. If it is HR, then HR denotes for lowercase. Then it differentiates the C and E based on the prime. If it's single prime, then lowercase c. If it's double prime, then lowercase e. So I repeat, if it is RH, that codes for uppercase. If it is prime, then it is C uppercase. If it is double prime, then that is E uppercase. If it is HR, it denotes lowercase. If it's prime, it's lowercase c. If it's double prime, then it is lowercase e. So important notations to remember in the transcription of your RH nomenclature. The presence of a superscript or a single prime that is denoting or the notation is for uppercase C. For double prime, that is uppercase E. Presence of subscript 1 would be for uppercase C. Presence of subscript 2, that would be for uppercase E. A presence of subscript 0, that would denote that both the C and the E antigens are in the lower case. Presence of subscript Z would mean that both the C and the E antigens are in the uppercase. Absence of 1 or single prime would denote a lowercase c. Absence of double prime or 2 would denote a lowercase e. A lowercase r would denote a lowercase d, while an uppercase r would denote an uppercase d. So this one would mean Rh negative, and this one would mean Rh positive. So let's do example exercises. So let's see. This is an uppercase R with subscript 1. So how do we transcribe it in the Fisher race? Okay, Fisher race terminology is DCE. So let us look at first at the D. Is the D uppercase or lowercase? Since it is an uppercase R, so therefore you write uppercase D. How about for the C? That's the second. Is the C uppercase or lowercase? Look at the subscript. It is 1. It is said that 1 or a single prime would denote C in the uppercase. So therefore, we write uppercase C. How about for E? For E, is there the presence of double prime or 2? subscript 2, there's none. So therefore, it means to say that the E antigen would be in the lowercase. So this is the Fisher-Race DCE. If you want to transcribe it in the CDE, in alphabetical order, this would be, oh, this should be an uppercase D. Correction. This should be uppercase D. Ito siya. Okay? So that should be uppercase C, uppercase D, lowercase e. Okay, next, this one, lowercase r. For lowercase r, sa DCE Fisher race mo, look at first at the D. D, it's lowercase r, so therefore lowercase d. It's RH negative. How about for the C? There is absence. Wala kang nakikitang single prime or 1. So therefore, lowercase c. How about for E? You don't see double prime or 2. So that means that codes for a lowercase e. So a notation of a single lowercase r would mean you have lowercase for both DCE. This is for the Fisher race. If you're going to use the alphabetical order transcription, then it would be CDE, all in lowercase. Okay. Third example, you have here lowercase r with a superscript double prime. So start Fisher race, start with D. It's a lowercase r, so therefore the D should be lowercase. How about for C? There is absence of single prime and 1. Wala ka namang nakikitang single prime or 1 dito sa baba. So therefore, it means that the C is in the lowercase. How about for the E? You see double prime. Okay, walang 2 but there is double prime. So meaning to say that your E antigen would be uppercase. So if we do the alphabetical order, this would be the transcription C, D, E. How about this one? You have uppercase R, subscript 0. Okay. So start with the R in the start with the D in the DCE Fisher race nomenclature. D would be in uppercase because this is uppercase R. But you have subscript 0. What the subscript 0 denotes? It denotes that both the C and the E antigen would be in the lowercase. So you have DCE. If you will be following the alphabetical order transcription, then this would be the C, D, E, only the D in the uppercase. So I hope the examples is very clear. And once again, I would like to emphasize you have a error here, technical error. Itong D dapat supposed to be uppercase dahil uppercase R ito. Okay. Another emphasis, if you're using 1, 2, 0, or Z, it should be subscript. If you're using the prime, double prime, it should be superscript. Okay. Next, the Rosenfeld nomenclature. Antigens are designated by number, where uppercase D would be 1, uppercase C would be 2, uppercase E would be 3, lowercase c would be 4, lowercase e would be 5. Okay. We have an example. D positive, uppercase C positive, uppercase E negative, lowercase c positive, lowercase e positive. So we transcribe it in Rosenfeld. RH would be +1, +2, -3 tayo because your uppercase E is negative. Then +4, +5 because they're all present. So except for the E, which is noted for number 3, yung notation niya will be negative or -3 since the uppercase E is negative in the example. Let's take a look some other examples. So you have here a genotype. Ito ating genotype because they are codominant genes. So tatlong makikita. Ito is one haplotype, another haplotype, and all this would be your genotype. So let us take a look. RH. What about for the uppercase D? Is it present? Yes. So you have positive 1. How about uppercase C? Yes, there is. So you have positive 2. How about uppercase E? There's none. So you have -3. How about lowercase c? Yes, there is. So you have positive 4. How about lowercase e? Yes, you have to both haplotypes. Then you have positive 5. Second example, let us take a look. The RH would be, for number 1, it's present in one haplotype, so positive 1. How about for uppercase C? Present to both haplotypes, you have positive 2. For number 3, yes, meron ka both nga eh, no? So plus 3. How about lowercase c? Wala. Kahit in either side, wala. So that's negative 4 or minus 4. How about for the e na lowercase? Wala rin. So you have minus 5. Okay. How about the third sample? RH natin, it's negative 1 because you do not have the uppercase D. So this is an RH negative. For the uppercase C, minus 2 rin, minus 2 dahil wala. It's not present. For uppercase E, yes, to both haplotypes. So you have plus 3 or positive 3. How about for lowercase c? Yes, to both haplotypes. So that is positive 4. How about for the lowercase e? Wala in either haplotype. So therefore, it is negative 5. Okay. Genotype versus phenotype. The phenotype is the result of the reaction between the red cells and anti-sera. So the one that we perform in the laboratory, serological testing, wherein we test the 2 to 3 or 3 to 5% of red blood cell suspension with your anti-D for the case of Rh typing is what we call the phenotyping. So whatever would be the result based on our interpretation, that would be the phenotype of the individual. While the genotype is the genetic makeup and can be predicted using the phenotype and by considering the race of an individual. And they are expressed in haplotypes, no? Dalawang haplotypes, tatlong genes nandoon because they are codominant allele genes. The CDE or the DCE terminology natin. So these are the genotype, ito yung symbol, and then your status. Again, yung status natin, titignan lang natin kung may presence or absence of the upper case D. So in this case, wala talaga, so therefore RH negative. Ito naman, meron ka isa. So even if there is one, this is a heterozygous positive. No? The uppercase is dominant over the lowercase. So this one is expressed. So whether it is a combination of upper and lowercase D, still it is regarded as D positive. So this is heterozygous D. How about this one? This is homozygous D kasi both are uppercase D. This one is heterozygous. This one is homozygous. This one is also homozygous. Okay, how about for the symbol? So, itong symbol na isa is for this haplotype. The other one naman is for this haplotype. So, CDE because you only have RR lang ito na lowercase. So, maintindihan naman. Let's take a look on the second example. Let's take this haplotype. The C mo is uppercase, D mo is uppercase, lowercase mo ang E. So, that's R1. No? R1 na uppercase R for the D. And then uppercase C for the 1. Lowercase e kasi, lowercase e siya. And then, when you transcribe it, wala ka absence of the double prime or 2. Dito naman sa kabila, kasi all are lowercase, pwede mo lagyan ng lowercase r lang. Okay? Kasi lowercase r would mean lowercase d. How about for the C and E? Wala naman. Absence of prime, double prime, or 1 or 2, then all will be lowercase. So the same is true sa other genotypes. Okay. So ito naman yung ating RH phenotype and transcribe it into, oh sorry, I will go back to that slide and transcribe it to the CDE terminology. Okay. So, transcribe lang natin. Okay, CDE. Sa C mo, anong C mo? So, sa first mo, may 1 ka, so that represents the uppercase. Sa second mo, absence of single prime or 1, so therefore lowercase. Ang D mo, tignan mo ang D, uppercase ang isusulat rather than the lowercase because uppercase is dominant over the lowercase. Remember, this is phenotype, so consider the dominancy. Yung kanina genotype yon, okay? So, isa lang isusulat for the D. Kung ano yung dominant yung ating isusulat. While for the E, there's absence of double prime at saka tawag nito at saka double prime or 2. So to both haplotypes, pareho lang E. So wag na tayo mag-E na lowercase kasi E would mean enough na yon na on both haplotypes, you only have one lowercase e. Okay, how about this one? Sa R1, R2 natin. Second natin ha. Sa second,
ano ang ating C kasi si DE ito. Unahin natin si C. Sa C mo, meron kang uppercase for this haplotype. And then, sa second haplotype mo is lowercase c. So, kaya you have upper and lowercase. How about for the D? D mo, all uppercase. No? Again, kung merong mang heterozygous na lowercase r, still ang isusulat yung uppercase D. How about for the E? Both will be present. Kasi on this haplotype, ang E mo dito is lowercase. For this haplotype, kasi may 2, ang E mo ngayon is uppercase. So that's why this would be the transcription. So this all throughout the same na yun sa baba. Okay, pareho lang ang principle of transcription. The importance of the RH system. After the A and B antigens, the D antigen is the most important red cell antigen in blood banking. The D antibody can cause transfusion reactions and HDFN for the reason that it is IgG in nature. As we've said, it significant immunoglobulin because it is reactive at body temperature, which is 37 degrees centigrade. That's why it can cause implicated in HDR. At the same time, it crosses the placental barrier, reason for it is implicated in HDFN. Okay, D-testing. Ano itong D-testing? Una sa lahat, let us review first, how do we do phenotyping in the laboratory? Ang phenotyping natin, tube method, we make use of that. So, ang kailangan mo lang is 3 to 5 or 2 to 5% and then your anti-D. 1 is to 2 lagi, one part of the antigen coming from your RBC suspension and 2 parts of the antibody coming from your anti-D. And then incubate that at 37 and then spin, no? Pwedeng hindi na mag-incubate, diretso tayo spin. Okay? And then, grade for the agglutination. So, yun ang ating procedure for the testing. Now, there are cases wherein an individual would be typed as weak D. Yung mga mahina ang reaction, weak D siya. What are the possible causes for weak D? Ito is yung sinasabi nating D variant. Ang nangyari is you have a very less receptor site or antigenic site for a weak D. Kung meron ka mang around 50 sites, just an example, if you have a 50 sites of antigenic sites for a natural of a standard D positive, pagdating sa DU, maybe you only have around 5 to 10. So, napaka-konti ng antigenic sites niya. So, therefore, there would also be less antibody to react, causing it to cause a weak agglutination. Another case of a very weak is the partial D. Ang partial D naman is may missing portion sa iyong antigenic site, but we cannot disregard the fact na kahit merong missing portion, they are still Rh positive. Same is true with the DU variant, yung sinabi ko, the first one, yung weak D mo, na kung saan you have less number of antigenic site, we cannot also disregard the fact na they are also D positive. So if we don't establish whether they are weak D or yung ating partial na missing D, we will mistype the patient as Rh negative, right? No, kung hindi natin ma-establish yun, dahil weak na ang reaction, di natin nakikita ang agglutination, we mistype them as D negative, no? Now, ano ang clinical significance of this? It is really very clinical, or if it's really very important clinically to establish whether a person is a DU or a weak D. DU, weak D, or the partial missing D. No? Lalong-lalo na, importante, if we're talking about donors. Okay, bakit? Kasi pag donor ka and you are mistypable being Rh negative, so therefore, yung dugo mo isasalin sa isang Rh negative recipient rin dahil yun ay na-mistype ka. So ano ang danger doon? Ang danger is that you are mistyped as Rh negative na supposed to be you are D positive. at isinalin ang dugo mo sa isang Rh negative, then you expose that recipient to the D antigen, although weak, to the D antigen of the donor. So kahit weak yan, still it can cause hemolytic transfusion reactions. See how important it is to establish that a donor variant, a partial partial missing D, weak reacting, kung ito'y donor, kailangan i-establish. If the case would be ang DU variant natin o ang weak reacting would be the recipient, no need to establish. Kasi ang mangyayari, pag DU variant or weak reacting ang ating patient, then that would be mistyped as Rh negative. Walang bearing yon. Kung Rh negative siya, so therefore ang hahanapin na natin na donor would also be Rh negative. Walang bearing kahit pa sa type ang recipient na Rh negative, supposed to be Rh positive siya, at sasalinan siya ng dugo ng Rh negative, there would be no problem because in the blood of the donor who is Rh negative, there's just simply the absence of the D antigen. So walang problema, walang exposure doon. Hindi naman isasalin ng dugo ng recipient dun sa donor. Di ba? Iyo, ang ating transfusion is the donor's blood transfuse to the donor or to the recipient, no? So, walang problema. So, I repeat, napaka-importante that we establish whether a person is a DU variant or a partial missing D antigen if the donor, if the person that we're talking about would be the donor. Kasi pag na-mistype ang donor as Rh negative, na supposed to be Rh positive siya, then that poses danger to the recipient who is really true Rh negative because the patient will be exposed to the D antigen, although weak reacting from the donor. Okay? But again, pag ang patient is the DU positive or siya ang weak, there's no need to establish na siya ay weak or DU. Kung na-mistype man na sa Rh negative, then that will not pose any harm at all since pag na-identify siya or na-mistype siya as Rh negative, the blood that we will be looking for donation would be an Rh negative also. So there would be no problem since a D negative individual would simply mean that it does not contain any D antigens. Okay? So itong D-testing na ito is particularly to identify yung ating donor na possibly na mistype natin. So what do we do? So we add anti-D to the D tube and then we also run an RH control and then spin ito and record. Okay? We add 22% of albumin. The very purpose of this is to reduce the zeta potential. The same action with the enzyme that it will dissipate the electrical charges and so to prevent the red cell from being pulled apart from each other with the use of your 22% albumin that reduces the zeta potential by getting the cells nearer to each other or coming closer to each other. That's why no need for an extended time of incubation to allow sensitization. Malapit na sila sa isa't isa. And so therefore, using the LISs or your albumin, 22.2% that reduces the incubation time. LISS that reduces around 10 to 15 minutes, while for your 22% albumin would take around 20 minutes. For the standard NSS, say you're going to incubate it at 30 minutes for 30 minutes at 37 degrees centigrade. So we need to spin, read, and record. We wash it 3 times in saline. The purpose of washing is to remove the unagglutinated IgG, yung mga free na immunoglobulin na hindi nakapag-react to its specific antigen kasi nga hindi sila yung target antibody IgG. So we need to remove them. In order to in order to remove them because they can neutralize the AHG. No? So after washing, we add the AHG, the anti-human globulin. Ito yung anti-human globulin ninyo in my discussion in the introduction, that this would span the distance of an immunoglobulin G antibody because of its short size, around 14 nanometer, to which the standard distance between one red cell to the other would be around 24 nanometers. So therefore, if it's an IgG, it can coat the cells but cannot cause agglutination since cells are far from each other. So you need a reagent that will span the distance to attach to one antigen antibody complex to another until it forms the lattice, and so that's how it enhances the agglutination. Okay. And after this, if D is positive after heat and albumin or AHG, cells are weak D positive. If negative, cells are RH negative. The control, meaning the C, should always be negative. Now, what are we going to do? If it is positive indeed, then let it be recorded as RH, as negative, as rather positive. To all negative tubes, we need to check, no? Using the Coombs check cells, okay? Add check cells to negative tube, spin, read, and record. Okay, so we add check cells to all negative tubes. Why do we need to check? This is to validate whether a negative result is really a negative result. So lahat. Kasi kung negative result yan, ibig sabihin, the AHG that you have added are now free in the test mixture. Hindi siya nakapag-react, diba? So free ito siya. So, kung dagdag natin ang check cells, the check cells are there to attach to the free AHG na hindi na igamit, no? Kasi nga, nag-negative yun, so free sila. So, these check cells will attach to AHG to validate whether your result is really a true negative or not. Okay. Now, so what do you expect if it is a true negative? Pagdagdag ng check cells, should be an agglutination. Bakit? Because your check cells reacted to AHG na free in the test mixture. Okay. Now, if there would be agglutination upon the addition of check cells, then that makes a negative result valid. Pero if after adding the check cells, there would be no agglutination, then you have to repeat the procedure because your test now is invalid. Why is it so? Bakit hindi nag-agglutinate? Kung talagang negative yung result niya, supposed to be the AHG would be free in the test mixture. Pero walang agglutination. So anong possible reasons why um, invalidate yung ating result na negative? Una is yung incomplete washing. Sabi ko kanina, the washing will eliminate lahat ng mga unagglutinated IgG. Kasi pag hindi na itanggal ito, the AHG is specific to IgG. So kung hindi ito na itanggal yung mga interfering immunoglobulins G, then that can neutralize your AHG. So when you add the check cells, wala nang magre-react sa check cells mo dahil nakapag-react doon sa iyong unagglutinated IgG. So one factor would be incomplete washing. Pangalawang factor is maybe you really forgot to add the AHG, no? Hindi mo na idagdag ang AHG. So therefore, kahit i-add mo ang check cells, wala naman talagang AHG to bind in the first place. Okay? So again, if upon the addition of check cells, there would be no agglutination to a negative result, then you need to repeat that. Okay? So this is only done, again, this is only done the testing for a person who is possibly mistyped to be RH negative kasi siya ay DU, and what we're talking here, person as donor and not as recipient. Okay. So that would be all for our RH blood group system.